Hello, everybody. My name is Rajan Sampat, and I'm a junior here at Oglethorpe University. We're here to talk about the mRNA vaccine development for SARS-CoV-2. So I kind of arrived at this topic based on some of my interests in the time period we're in. Right now, we are in the pandemic. We've been in it for over a year now. And when we first got sent home from school and everything changed, there's always this talk of where are we in the vaccine stage? Is there going to be a vaccine in November, December? What's the timeline looking like? The race for a vaccine. This all kind of piqued my interest because we were hearing about it so much. It wasn't so much about the technology, but the timeline. When are we going to be expecting a vaccine? When can we expect to have it? So all this different stuff was being thrown around. And I was kind of interested to see what exactly was happening behind the scenes, like what is going into these vaccines. So with that, I'm going to outline my presentation. And so I'm going to introduce the vaccine technology, kind of give a little background on what exactly is happening. And I'm going to go into the SARS-CoV-2 infection cycle, or COVID-19's infection cycle, how it infects cells and what is occurring. And then I'm going to go into some previous research done on some other strains of coronavirus and see how that could potentially play out for a vaccine. And then I'm going to talk about the study that was done and the data that they have based on what they saw in their results. And then I'm going to wrap up the presentation with a couple take-home points, and we'll go from there. So to start it, I'm going to be talking about the vaccine technology. It's either nucleic-based or protein-based vaccines. So a couple of factors come into play here. It's either the production time and effectiveness. So protein-based vaccines actually take a while to develop. They take at least, sometimes in a case, a year, sometimes longer, whereas nucleic acid-based vaccines can be done in a very short amount of time, in a matter of weeks. Another thing that has been a big topic with vaccines is how effective is it, the efficacy rate. So nucleic acid vaccines have been proven to be more effective, or seen to be more effective than the protein-based vaccines. With that, they elicit immunity. So there are two types of immunity that can come through: humoral immunity or cell-mediated, otherwise known as cellular immunity. So humoral immunity is the idea that it's more of a short-term memory type immunity. It happens extracellular or outside the cell, and it is more of a B cell response with a little bit of help from a T helper 2 cell, but it's mainly the B cells that respond. Cell-mediated or cellular immunity is more of a long-term memory type, and it's intracellular. So this happens inside the cell. So if the virus infects a cell, this infected cell now is going to be taken care of by the immune system, and it creates a cellular immunity. So these cells are T cells, phagocytes, or even the release of cytokines are involved. So based on that, the nucleic acid-based vaccines were the way to go. And knowing that it could go DNA or RNA, DNA-based vaccines have been around a little bit longer, and mRNA vaccines are the newer technology, but have been so far seen to be more effective and can not only elicit humoral immunity, which most vaccines do, but mRNA vaccines can also elicit this cellular immunity as well. So with that, what exactly are these vaccines going to be targeting, and how is COVID-19 or SARS-CoV-2 affecting these healthy cells? So cells are getting infected because of the SARS-CoV-2 spike protein or S protein and its ability to bind with this ACE2 receptor on the plasma membrane. So on this demographic on the right, This is kind of the process of how it happens. So first you see the spike protein here, and it will bind with the ACE2 receptor, and eventually it will lead into the fusion of the two into the cell membrane. From there, the viral cell will be able to infect the healthy cell and continue to replicate. This picture on the right or the left shows the COVID-19 or SARS-CoV-2 spike protein and how it interacts with the ACE2 receptor and is able to bind and ultimately end up in this fusion state. So what exactly is ACE2? What is the receptor for? So ACE2 is the angiotensin converting enzyme 2. This is responsible for lowering blood pressure within the body, and these receptors are commonly found in the respiratory tract within the lungs, but can also be found in the heart, the arteries, the kidneys, and the intestines. So if someone were to contract SARS-CoV-2, not only is the respiratory tract exposed, but other systems in the body are exposed and are at risk. So COVID-19 specifically could be multiplying so rapidly and causing so much infection and the high infection rates because of its ability and the high affinity it has for seeking out these ACE2 receptors. So with that, there's been a potential way for the vaccine to be developed. There's been more research done on other strains of COVID-19, of coronavirus, and this could be responsible for a potential vaccine. So here, I'm going to take a quick look at this picture here on the right, and this is going to highlight that spike protein, the one that is able to bind with the ACE2 receptors. So the research that has been done on vaccines initially started on the MERS-CoV, or otherwise known as the Middle East Respiratory Syndrome coronavirus. And vaccine developed uses this MERS-CoV S2P. And that stands for the 2-proline switch that's actually seen in the central helix and the heptad repeat 1. So this heptad repeat, there's heptad repeat 1 and heptad repeat 2, but these are responsible for the ability of the spike protein to bind with the ACE2 receptors or receptors in general. So this helps with the ability to bind and multiply and fuse with the membranes. So this 2-proline switch is able to catch this spike protein in the pre-fusion state before it is having the ability to bind to these receptors. It is delivered in an mRNA vaccine that has LNPs or lipid nanoparticles. So these lipid nanoparticles actually are one of the more chemically advanced ways of being able to deliver these genes. mRNA was picked because it is a more efficient way of delivering the vaccine. So in mRNA, in a vaccine, it is injected into the deltoid, or for the most part, for these vaccines. And when that happens, the cells will take in the LNPs, or the lipid nanoparticles. With that, it will leave the mRNA in the cytoplasm, where it can be translated into the protein. If it were to be a DNA base, the DNA would have to undergo transcription, to become mRNA before it could go to the lipid nanoparticles. So knowing that mRNA is a little bit more efficient because it doesn't have to go to the nucleus to undergo transcription. And in this research of the MERS-CoV S2P, they saw that it was more immunogenic than the wild type MERS-CoV mRNA. So immunogenic is talking about how the immune response is to an antigen. So this is saying that basically the MERS-CoV S2P strain, the 2-proline switch, is able to create more of an immune response than the wild type was. So this now was being able to be taken, developed into the vaccine. They had the backbone for it and were able to roll it out. Now they were able to take this technique and use it with the human coronavirus and SARS-CoV and create vaccines for that as well. So with that, there's hope that you can take this technology or technique where you incorporate this 2-proline switch and create a vaccine and expedite the process for the SARS-CoV-2. So this paper here in the study that I'm going to talk about today is actually looking at that. They're going to look at the 2-proline switch and see if the delivery with the mRNA and the lipid nanoparticles will actually expedite the process and still provide an immunogenic response or a protective response. So the hypothesis here is if the mRNA and lipid nanoparticles delivery of the SARS-CoV-2 S2P is effective, then the mice should show an immunogenic or protective response against the SARS-CoV-2 strain. So first off, to begin, the methodology here is going to be for an antibody response. So these mice are going to be 6-week-old female mice, and they're going to receive 2 doses of the vaccine or a placebo 3 weeks apart. So this first dose is going to be the prime dose. The second dose is going to be the boost. And the dosages of the vaccine will be 0.01, 0.1, and 1 microgram. Every 2 weeks after the vaccine, so 2 weeks after the prime dose and 2 weeks after the boost, data will be collected to determine the antibody response and how effective the vaccines were. So looking at that, the mice were then undergone study and blood was taken to determine the antibody response. So to kind of get acclimated to the graphs here, the x-axis will show the dosages in either the placebo or the vaccine in both of these graphs. The y-axis is going to show the response in the blood. So the reciprocal serum here is going to show how effective it was in producing a binding antibody response in this graph. And then in graph D, it's going to show the titer that will help with a neutralizing response. So with that, the prime dose will be shown with these white circles here throughout in this first bar, and the second bar with the dark circles, the black circles, will be the boost. So originally here in the binding antibodies, after the first dose, the prime dose, the placebo 0.01 and 0.1 microgram doses didn't show very much efficacy or production of the antibodies. Whereas in the 1 microgram dose, there was some production of it. So that shows that they had some sort of protection after that first dose. Whereas in the second dose, in the binding antibodies, there was almost a double in the amount of antibodies that were available in the 1 microgram dose. And there's some sort of protection here, as there was some sort of protection in the 0.1 and 0.01 after the boost. In the neutralizing antibodies, the 0.1 and PBS showed very little, if not any, response. But the 1 microgram dose was able to show neutralizing antibodies, which means the vaccine was able to help prevent or inhibit the growth of the virus or replication, while also showing that it can produce antibodies that can help with recognition and eliminating the binding of the ACE2 receptors. So with that, now that this has been recognized, they also want to test this vaccine in the case of being infected. So that is exactly what they have done. So in the methodology for determining the effectiveness here, the mice were just young adults. They either got 1 dose or 2 doses of the vaccine. Those that got 2 doses either received dosages of 0.1, 0.01, or 1 microgram doses. Those that received 1 dose got 0.1, 1, and 10 microgram doses, one of the 3. After 5 to 13 weeks, they were challenged with a mouse-adapted SARS-CoV-2 strain. And this means that the CoV-2 strain had a switch in the receptor binding domain, which would allow for the SARS-CoV-2 to be able to bind to the ACE2 receptors in the mice, which would allow for the respiratory tract to be involved. So then actually 2 days after the challenge, data was collected, and lung tissue was also collected to be imaged. And then they re-imaged lung tissue 4 days after the challenge or the exposure to COVID-19, SARS-CoV-2. So with that, the mice here were organized into either 2 doses of the vaccine, and these are the doses that they received, along with a peak viral load that was seen, or they got 1 dose, and the dose is seen here below at the 0.11 microgram or 10 micrograms, and the peak viral load here. So with that, the placebo here, or the control group, had a rather high peak viral load, and it's seen that 0.01 is still high up there, but it's a little bit less, and it continues to decrease as you work down the 2 doses graph. So you see that 0.1 had a decrease, and then 1 microgram with 2 doses of it showed that there was very little, if any, viral loads to be found. When you look at the 1 dose, the 0.1 was not very effective, and it has a same viral load that's close to the control group seen in the 2 dose, whereas the 1 microgram and 10 micrograms proved to be rather effective. So the mouse tissue here shown has the day 2 images here, and then day 4 images here. This top row is the control group. The middle row is 2 doses of the 0.1 microgram vaccine, and the bottom row is 1 dose at 0.1 micrograms of the vaccine. So in 0.4, it's kind of hard to see some of these things. I know in the control, you can kind of see some areas of swelling or where it's a little bit darker. And when they go to the 10x magnification, it shows that there's more inflammation, it's more severe, like moderate to severe. And that actually in day 4, there's less of the, there's less alveolar space. So that signifies that there's more inflammation, there's going to be more effect here. In the 2 doses of the 0.1 micrograms of the vaccine, the mice themselves exhibited very little, if any, swelling, and it's hard to pick up. But these darker spots right here are kind of where they show some swelling. And even then, even at day 4, there's still very little swelling. There isn't very much. And the inflammation is seen in the bronchioles and the bronchovascular bundles. At the 1 dose of the 0.1 micrograms, there's more space in the alveolar septae, and this will be responsible for the cause of some inflammation, which is seen here in day 4, where there's more inflammation. There's like mild inflammation seen. So while the mice were exposed to a variety of different doses, this kind of goes along with the idea that the vaccine is very much dose dependent. At the lowest doses, there's still some sort of protection offered for the mice. And these mice at the ideal dose are almost always protected. So to kind of wrap it all up, mRNA vaccines are the future. There have been some small sample sizes seen with the coronavirus vaccines, but they have been proven to show that they have more potent immune responses, and they're able to produce some sort of protection for an individual or an organism at even the smallest doses. Like even at the 0.01 micrograms, there was still some sort of protection. Prior research has really helped with the development of the SARS-CoV-2 vaccine. The vaccine itself was developed in under a year, and the original MERS vaccine took about 22 months to develop. So this prior research has really helped expedite the process and speed up where the vaccine is gone. It's been very important and critical to see the research that was done and how it is applied to these vaccines. And actually, the mRNA 1273 vaccine that was used in the study was Moderna's vaccine that is now open to the public to get here in the state of Georgia. This is one of two vaccines that are available. Of course, it's the other one being Pfizer. And this means that they have data that not only shows that in organisms, it can prove that to be beneficial, but also in clinical trials. In clinical trials, they had a 94.1 efficacy rate, which is outstanding considering that some of these vaccines for respiratory illnesses, such as even influenza, an inactivated vaccine has as low as like 60%, I believe. I saw. And so Moderna and Pfizer were able to accomplish a lot. It just shows that there is a lot of research, and part of this attributes to the idea that the prior research on the 2-proline switch really helped them expedite the process and build a safe and effective vaccine. I would like to thank you guys for watching my video, and I really hope you guys enjoyed it.